So our next speaker, kicking off the second half, is Ian Baker from Colorado State University. I've known Ian for a long time. He's a great ecosystem modeler, really looking at the global scale, and he works on the model SIB, which has been mentioned both by Joe and Elliot earlier. And there's multiple versions of SIB that he's helped develop with his team, both in Colorado and worldwide. What's nice about Ian is that he really tackles kind of all the hot topics, and whether it's in the tropics or the Arctic, whether it's with fluorescence or OCS, new data sets, he's always putting the modeling efforts into confronting, developing, and understanding the models, not afraid to show bad model results, and I really appreciate that because that's where I feel like we can learn the most. Oftentimes when modelers really try to, as Elliot said, force to a common number, are you really learning anything about the process and how it works in a model? And I think Ian really exemplifies a leader and a great communicator in understanding these connections in the complexity of these models. Ian.
Thanks. This is going to be a fun week. I'm looking forward to this. A couple of points before I begin. First is, my name's on this, but this is collaborative work with a huge group of people, a lot of them in this room. I'm fortunate enough to know smart people, and I basically get their research and we say, well, let's put it in the model and see what happens. And so I'm fortunate to be able to work with a lot of people who have very interesting science that we try to incorporate into our models. A second one is, I say OCS. And the reason I do is because I'm also I program computer code. And so COS is for cosine. So OCS in my code, and so that's the convention that I use. And then and another, you're going to notice during my talk some things, I don't want to use the word redundant, I think complementary to some of the things that Joe and Elliot were showing. You're going to see a little bit of, again, some of it will be a little, I don't want to use the repetitive. It's not going to be repetitive, it's going to add to things that they've already said. So a little bit of background, you've seen some of this. This is from Mauna Loa, right? We know that CO2 levels in the atmosphere are going up through time. You can also see the mean signal is shown in black, and then in red you can sort of see the breathing of the northern hemisphere, because that's where the land is and all the plants. And you have these little sort of minor drawdowns in the boreal summer and then a little increase in the boreal winter. And another thing we know is we know from economic data approximately how much CO2 we put into the air each year. This is sort of old data. It only goes up to about 2010 or so. This number is actually up around 10 now. And so we know how much we're adding to the atmosphere each year from economic data. We know how much CO2 is in the air because there are sites now not only at Mauna Loa but around the globe. And so then we can sort of subtract these two, and we know what the retention amount is each year in the atmosphere, and it's only about half. And it changes from here. Here's 1998, when almost all the CO2 stayed in the atmosphere that year. Here's 1991, not very much. And you see quite a bit of variability. And this variability is a result of a small net signal in large gross fluxes. Joe, I think, talked about this a little bit, sort of the average accepted from some estimates is about 120 gigatons, both coming into the land and then coming out of the land into the atmosphere as plants die and little microbes respire the CO2 out of them. You have a similar magnitude signal in the ocean. And then you have this 10 gigatons per year coming out from anthropogenic, and you're seeing about half of that being taken up by the biosphere, about half of that half in the ocean, and then another little part on the land. And one of the things that we know is that most of this variability comes from the land. There's a lot about that we don't know. We don't know where it is. Is it in tropical Amazon? Is it boreal forests? We don't know exactly what causes the interannual changes. There's some link to El Nino. I'm not going to talk about that today. And another thing we don't know is about the future, and Elliot sort of touched on this a little bit. We've got about this half of our anthropogenic CO2 is being taken up each year. Is that going to continue? Is it going to increase? Is it going to decrease? Is it going to stop? Is it going to switch signs? And those are questions that we don't know the answers to right now. And so right now we can say that plants are growing faster than they're dying. This is a paper that's showing NPP, net primary production, which is the total photosynthesis minus the respiration from the plant itself. And so what you see is that right now what's going on is the plants are growing a little bit faster than they're dying and being decomposed. Some of the reasons might be CO2 fertilization. If you have more CO2 in the atmosphere, plants take CO2 up through the stomates to make starches and sugars and leaves and wood. For the same stomatal opening and higher CO2 in the atmosphere, you can get more CO2 into the plant. Some regions might actually get a little nitrogen fertilization from sort of industrial pollution can actually work as a fertilization. You have woody encroachment. If you think about far northern regions where you transition from maybe a forest into the tundra, that forest might be growing a little bit further north. You might also have a longer growing season in some of those regions where maybe at one point your growing season was May to September. Now it's from mid-April to mid-October. And that's just more time that you can take up CO2. In the US, especially, fire suppression has been mentioned. If you're putting out the forest fires, then you can accumulate this woody material on the ground. And again, we don't know how these things are going to behave in the future. We don't have, we're seeing this signal right now, but we don't have strong, physically based, process-based ability to predict what's going to happen in the future. So we're trying to quantify this sink that we see, right? We have this small sink between these large gross fluxes, but you can't just put it in the model. It has to be emergent in your model. And what that means is I can write a model that says there's 5 gigatons of sink in New Jersey if I want, right? It's a model. I can do whatever I want. But what you need to have is you need to have a model that you then use in what we call inversions. And I think inversions might have been mentioned. I'm not going to talk too much about it, but just to say that what you'll do in an inversion is you will write a model of these sorts of fluxes. And you will then run them, right? You'll have your land taking up and releasing CO2, your oceans doing the same, you'll have some industrial sources, you'll have a model, a transport model blowing the wind around, and then you take observations. A lot of times it'll be from flasks from around the world, and you do some matrix algebra, and your inversion will tell you where your initial model ideas are wrong. And so what we're really doing is we're trying to use these models as sort of these a priori ideas of where and how things are happening here, and then the observations will inform us on where we're right and where we're wrong. And so just a brief chat about some of these models, and really we're going to talk about okay, wait, that's next. The first thing is to say, and you've seen this a couple of times, is do our models agree with each other? This is now boreal North America, you can think Canada. This is temperate, you can think United States. Here's an annual cycle and then a whole laundry list of models here. And what you see is that these models differ by a factor of about 2. Joe showed this, I think Elliot had some of this as well. And so our models do not agree on the amplitude. And Elliot actually said that, right, you can have these models with these large differences in their gross fluxes, but they can add up to get a similar net. But the idea here is as long as you have this spread, what it means is we really don't have a good handle on the mechanisms that are going on the response to variability, not only at the diurnal, but the synoptic and longer scales as well. So we can talk briefly about some types of models. This is a sort of a schematic of what happens, what are called a light response model. So this is light amount on the x-axis. You don't even need to know units here. And what you see is, right, photosynthesis increases rapidly at lower levels of light and then sort of asymptotes out. These are very simple statistical models. You have a few parameters, a few equations. You might have vegetation type as one of your parameters. You might have a response of photosynthesis to temperature, a response to drying out of the soil. And you're really combining multiple physical mechanisms into a few equations. CASA, that was in Tim's paper, is a light response model, and it's a very successful light response model. I work with a model called the simple Biosphere model, and this is what's called an enzyme kinetic model. These are much more complicated models. This is a schematic. You don't really need to know everything that's going on here. But the idea is that we have a much more complex and an explicit representation of physical processes. So we will have a parameterization of soil microbes, respiring dead carbon. We will have regulation across the stomate. We have C3 and C4 photosynthesis. We have radiative transfer through the canopy, take-up of water by roots. And we really will then need a lot more parameters. We will need parameters not only for vegetation type, we'll need parameters about the leaf properties, not only leaf angle distributions, but their transmissivity and reflectivity. We will need root distributions. How many roots do you have at the surface? How do they decrease with depth? And so people who prefer light response models will say that you have this over-parameterized behemoth. You have processes that you don't understand. You have parameters that you don't know the values of everywhere. And so that really sort of is detrimental to your ability to get a good physical representation. And they're right. And someone like me can go back and say to their light response model, I can say, you have a model that sees the sun rise and set. And I'm right too. And what you need to understand is all models are wrong, but some models are useful. And so I'm going to make the case that I think that in particularly with regards to SIF and carbonyl sulfide, with a process-based model, I'm better positioned to use this new information. I've got a process-based model. Joe or Roisin or Mary can give me some equations, and I have a place to put them in. And with a light response model, I'm not saying it's impossible, but it's going to be harder to do. And so that's one of the reasons why I like this kind of model. But you also do have to recognize that there are limitations, right? We don't know the values of all the parameters everywhere, but we have an ability, I think, with this kind of model to do maybe a little bit more exploration, a little bit more detailed exploration. So what I'm going to do today is briefly talk about just a couple of places where we've used both carbonyl sulfide and later on fluorescence in our models. So what we've been able to do is, Joe showed that schematic. I think I actually have it next. Yeah, he showed the schematic of the leaf here and how carbonyl sulfide is taken up through the stomate. We were able to take those equations and put them into our model. And so we've done a variety of studies where we sort of explore that behavior. And what I'm going to show you is a couple of spots where we've had it applied at a tower site, and then a regional application as well. So this first one, this is this paper, the Berry et al. 2013, and this was really the first time that anyone had tried to do sort of an explicit physically based representation of carbonyl sulfide. And one of the main results of that paper was that we said to balance the amount of plant uptake that we're getting in our model, we had to have this massive extra ocean flux to balance it. Otherwise, we would have sucked all the OCS out of the atmosphere. And Roisin disagrees with me a little bit, but I'm going to show this anyway. Subsequent studies, both from satellite and some inversions, as well as aircraft observations, have shown that there is this source in the ocean. Joe showed it in that animation that he showed at the end of his talk. So in this case, our model said, we're seeing something that has not been observed previously. Here's what we think is happening. And subsequently, a number of observations have, if not proven it, they have certainly supported our finding.
Here's,
yeah.
Can you just summarize briefly why someone might have a problem with this?
No, I think that's going to happen later in the week.
Just saying, some people have a problem with this.
I know that. What I will say is, I will leave that to the smart people to hammer out. And when they do, I will put their equations into the model and we'll see. Another thing that I say is, I know just enough chemistry to get myself in trouble. So the short answer is I'm not going to go there. Our model said it had to be that way. I'm sorry, this is a little bit washed out. This is these are some model results. Linda Kumans from she's in Groningen, but some studies up in Huchelen. This is a pine forest, and what we're looking at here is CO2 uptake over a day, the diurnal cycle, and carbonyl sulfide as well. And the model is in red and the observations are in blue. And what you see in each of these cases is our model is not taking up as much CO2 or OCS as the observations suggest. But if you look at the leaf relative uptake, and leaf relative uptake is the basic relationship between OCS and CO2 flux, you have to put a concentration term in there. But what you see is that our leaf relative uptake in the model is actually doing quite a good job here. Not only at nighttime, remember you can still have OCS uptake at nighttime when stomates aren't fully closed, but also during the day, and not only the overall, but you see a change during the day from sort of higher values in the morning and later afternoon down to a little bit lower value in midday. Elliott showed some models where the leaf relative uptake is actually imposed on the model. In our simulations, it's an emergent property. We calculate the physical processes, and then you can calculate a leaf relative uptake based on what the model does. And in this case, what it's suggesting is our model is getting the wrong answers, but it's getting them for the right reasons. And so this now informs us on ways that we might improve our model. In this case, what it makes us think is how we're specifying our vegetation, our canopy, is probably, we probably don't have enough leaves. And so research is ongoing on exploring how we can now change, we can use the information that we have to improve these simulations and try and get the right answer for the right reasons.
Sorry, quick question. When is sundown in that, roughly? OCS uptake is extending into the night?
Yeah, so at sundown, photosynthesis is going to stop because the sun's gone down. But both in the model and what people have informed me is that the stomates don't close completely at night. And so you can still have a little bit of uptake during the night. And in fact, what we see is as you get the nocturnal layer develop at night, right, the air will get very stable and you will actually deplete your CO2 during the night, or not your CO2, your OCS at night as it's taken up through the stomates. I mean, they're not wide open, but there is a little bit dribbling in. So this is now from a paper that Roisin did a couple of years ago where we did simulations at Harvard Forest. And what we're seeing is now the growing season signal. This is for daytime and then this is for nighttime. The model's in red. And what you see is we do a reasonably good job during the daytime. At nighttime, we're maybe not drawing up as much as we think that we should in some of these cases. In this case, we don't think that it's because our stomates aren't open enough. I actually think it's because when we calculate the nocturnal stable layer at the top of the canopy, we actually get almost a material surface in the model. You get no exchange at all. Whereas in the real world, if you're in Harvard Forest or some forest at night, even though it's a fairly calm night, every now and again you'll get these little eddies come rolling through. And we don't really have that ability in our model. So this is another place where now these observations, maybe this wasn't what we expected to find. This wasn't a place that, right, our turbulent transport and our stability calculation was not a place that we expected OCS to inform us about model shortcomings, sort of an added benefit. And another sort of the elephant in the room here is what the heck? You see now that there are places here where both at daytime and at night you have a positive value, an efflux from the biosphere, and Roisin tells me that she did her best to sort of find a way to an excuse to get rid of this, but there isn't one. And so this is real. And our model, as it is today, does not have a way to get a positive value. We have uptake of OCS into the stomates, we have uptake of OCS into the ground, and we do not have the ability to get a positive value. So this is certainly a place where we have to improve our models. And so that's one of the real reasons why I'm here this week, is to talk to smart people about what are some of the ways, what are some of the mechanisms by which this is happening, and how might we quantify those and put them in the model. This is now an inversion result that Hugh-Lin Chen and his team have done. And what we're looking at here is, this is now 2010, so you see an annual cycle on the x-axis. We see concentrations on the y-axis. So what we've done here is we've put our model fluxes into a transport model, and then we calculate how much CO2 or OCS exists. This is at a site in Iowa. And so what we have here is we have an observation in blue. We have a prior. This is our first guess of the model. And then we have the optimization. This is the one, remember I said you do some matrix algebra, and it tells you what it thinks your answer should be. And so in this case, if you look for CO2, the observation and the prior are actually quite close together. But for OCS, our prior is showing much, much more drawdown in Iowa, in the crop region, than the observations suggest. So in this case, what the inversion did was, it had to move our OCS up to get this optimized value, but in doing so, it also decreased our drawdown of CO2. Now, I've been doing or been involved with inversion studies for a decade at least, and I have never ever seen us have not enough uptake in the crop regions. When I see this result, my suspicion meter just redlines, it pegs. So what this says to me is this might be another region. There's been actually quite a bit of work that suggests that in heavily fertilized areas, you can get an OCS efflux from the soil. Again, we're just taking it up. And so this is another place where now the inversion is suggesting, and again, it's about that soil and or ecosystem efflux capability where our model is deficient. A small aside here in the paper, in Tim's paper that Elliot was showing, where SIB, Simple Biosphere model, didn't have very much uptake in the crop region, that was actually a different version of the model. We have Randy Locupiccia and co-authors actually wrote a SIB crop module that we're now using, and that gives us much stronger uptake in the crop regions, and it's actually quite realistic. So if we were to run again in Tim's setup, we would probably get a little bit different result with SIB. I'm hoping that we would look better in that area where we were looking bad, but we'll see. This is part of the fun of it. So I think now I'm going to switch to a couple of SIF studies.
Can you just, isn't there enough, Mary, you have a paper, from the agricultural.
Oh yeah, and you put the model you put my soil model into SIF.
Yeah, we've actually done, I can show some of those later this week. We've done, so you sent me some equations that you had and some that Cadmiel had. And I put both of those into the model and ran it around, and we do have some differences in our soil term.
Empirical parameterizations or something?
Yeah.
Just to sort of get an idea of if you subtract this out.
Yeah, that's
Yeah.
that loose model in there, right?
That's right.
That's the plan.
That's right. Yeah, and so this is I think you're gonna, you smart people are gonna have me bugging you about this all week, so be ready. So now I'm going to talk about a couple of SIF studies. So we have put SIF into SIB. Our SIF is not the same as SCOPE, which is Christian van der Tol's model that a number of you will be familiar with. It's similar, but not identical. So I'm just going to talk about a couple of regional studies, one in the Sahel and one in tropical South America. And I call this one the Goldilocks story. And I actually was talking about this with a European colleague the other day, and he didn't know the Goldilocks story. So I'm just guessing everyone knows Goldilocks and the Three Bears. The porridge is too hot, the porridge is too cold. I'm getting some nods and a couple of blank looks.
They don't have kids yet.
That must be it. Yeah, so Goldilocks and the Three Bears. Goldilocks walking through the woods, comes to the Three Bears' house. Porridge is the one porridge is too hot, one's too cold, one's just right. And she doesn't eat the ones that are too hot, too cold. She eats the ones that are just right, and then the bears chase her off into the woods. There's the condensed version. So first we're going to talk about the Sahel. This is, I'm going to show a few results from this site right here. This is a very seasonal region. The intertropical convergent zone, which is a zone of convection, moves north and south. And you see this is now the annual, an average annual cycle, and precipitation is in the bars. Temperature is in the dashed line, radiation is in the solid line. The main thing to take from this is you get very seasonal precipitation. Another thing about this is it's poorly observed. First, because there aren't many people in parts of this, or they're very poor countries, so you don't have a lot of observations. And also it can be politically unstable. A friend of mine had a tower up here in the Sahel, and then the Taliban took over the region. And so he couldn't go back to check on his instruments. And then I think the French came in and took the Taliban out, and he just had to leave it. So it's very poorly observed. So this is a place where SIF, where fluorescence from a satellite, might then give us some information that we might not have otherwise on behavior. Here's a plot similar to one that Joe showed, where we have GPP from the model on the x-axis, and we have observed fluorescence on the y-axis. And one of the things you see, okay, so this is 6 years of data, and I have labeled each monthly average with its number. So here you see July, here's June, right, and you have 6 of them for 6 years of data. August up here. And so what you see is, during the period when you get that onset of rains, you get a very rapid increase in the fluorescence. You also get an increase in the GPP. And then when things dry out, you can see here's September, October, November, you get a decrease in both GPP and fluorescence. And then here you have sort of your winter, your December, January, February, where things are pretty much dormant. And if you look at this, you say, hey, we're doing a pretty good job. We're getting, explaining 84% of the variability with our model. But I think we can do better, because now what we're doing here is we're looking at model photosynthesis against observed fluorescence, but I want to look at modeled fluorescence against observed fluorescence. And when we do that, the picture changes quite a bit. And so here now what you see is the same plot as we had before, but if the model was perfect, it would lie right on the 1-to-1 line. And it doesn't. You see a couple of things. First, in June, July, and August, the observed fluorescence increases more rapidly than the model fluorescence does. The other thing you notice is the amplitude of the signal. You go from almost 0, maybe 0.2, up to 2.3, 2.4 in the observations. And in the model, we go from about 0.5 to maybe 1.5. So the timing and the amplitude are different in the observed fluorescence than it is in the model. And this gives us an idea of maybe why we're being wrong here. And the reason is that we are using leaf area that comes from a satellite. This is MODIS-based leaf area, and MODIS has a problem with clouds. And so when you go to this time of the year, when the clouds show up and it starts raining and the grasses grow up quickly, the MODIS LAI-FPAR product doesn't really capture it. We get a very slow, very gradual uptake. And that's because MODIS has a hard time seeing through clouds. The wavelengths that it's looking in, it's hard to get a retrieval. Observations on the ground suggest that it's much more seasonal. It's almost bare during the dry months, and then as soon as it starts raining, bam, you get a signal that it really jumps up. And so the two differences here is, one is you've just sort of damped out with the MODIS product your seasonal cycle. You don't get the rapid uptake or the rapid decline in the grasses. So what we've been able to do is to use fluorescence data from OCO2 and soil moisture data from SMAP, which is the soil moisture active passive. Of course, the active part died, but we can still use the passive. And both of these are products that can actually see through clouds. They're able to retrieve through much thicker clouds. And so we're now able to observe this time of the year. And so we have a student who did a new model of phenology. So Kathy Haynes wrote a phenology model, and Dakota Smith tested it out. And what we have here now is surface soil moisture on the x-axis and fluorescence on the y-axis. And what you see, now we're only looking at the onset period, but what we're seeing is now when the rains show up, our model is now getting a rapid rise in fluorescence. And so we think that's making our model more realistic. Now what I'm not showing you on this is you notice that there are no numbers on the y-axis. And the reason is that our modeled fluorescence is much higher amplitude than observed. And so this is now suggesting another place where we have a problem. And I'm going to talk about that with people during the week, but I think that it has to do with how we calculate our photosynthesis in various quantities on a unit leaf scale, and then we do a leaf to canopy scaling. And we're thinking that our leaf to canopy scaling for photosynthesis might be inappropriate to use for fluorescence. But that's just another, I don't have an answer for the model here from that. This is just something that we're finding. But we are seeing that our timing now in response to moistening cycles is much better in terms of the sort of how quickly our model turns on and starts photosynthesizing. Finally, I'm going to get to my little Goldilocks story here. So this is a picture of the Amazon basin. What we have, you can't see the units on the scale here, but this is rainfall in meters. This is 3 meters or more up here. And what you see is that your precipitation amount declines as you move to the south and east. And this is now the dry season length, number of months with dry season or with precipitation less than 100 millimeters. And so you see that not only do you have more precipitation up here in the north and west, but the length of dry months increases as you move to the southeast. And this is really the defining characteristic of the ecosystem here, because it's always warm. You're right on the equator. There's always enough light for photosynthesis. You don't have the strong seasonality like you will maybe in boreal regions. And so this is really the defining characteristic of the ecosystem. And here's Scott Seleska's figure that I show all the time. There you go. I should get a residual from this. This is from an area near the Tapajos River where you see now a seasonal cycle. Here's precipitation on the bars, and then this is CO2 flux, where positive is into the atmosphere, negative is uptake. And what you see in the observations here is that you actually have an efflux of CO2 during the wet season and an uptake during the dry season. And then you have two models where basically they get exactly the opposite. If you multiply them by -1, you would get a better result. Now, I don't want to cast aspersions on anyone else's model because mine did the exact same thing. Here's now the seasonal cycle. You see the precipitation, you see the observed fluxes, you see the SIB. And so we had the exact same behavior. So in this case, we were able to say, okay, we hypothesize, here are some reasons why this might happen. We can put them in our model, and then we can run it again. And we did that, and now you see that we match the seasonal cycle. And so what we did was we said you have to have a deeper soil. You have to be able to store this water that you get during the wet season so you can use it during the dry season. And we also had to change how the model takes up water by roots. Most models have more roots near the surface, fewer roots with depth, and so if you dry out your surface soil, you're done. In nature, it's been observed that a few roots down deep in the soil can actually transport quite a bit of water. And so by incorporating those two mechanisms, we were able to match the observations at this one site. We then put that into the model and ran the entire region. And so now we can do a quick evaluation of how it responds to the 2010 drought. There were droughts, there have been droughts of the century in 2005, 2010, and 2015, but we're just going to look at where we're thinking we need to change the definition of drought of the century in the Amazon basin now. But this is one from 2010, and what you see is this area of water deficit sort of along the southern edge of the Amazon basin, the southern edge of the forest. And you can actually see that this is the GOM2 fluorescence, and you see that this is September 2010 subtracted from September 29. So a positive value means that the fluorescence was higher in 2009 than it was in 2010. There was a decrease in the fluorescent indicating this is where the drought was. We can look at it in our model and we don't see it. We do sort of capture this little area up here, but not really where the drought really hit in 2010. And so it appears that we have made our model too drought resilient. So this is where Goldilocks comes in. Before this paper that I showed from 2008, when we had the inverse of the annual cycle, our porage was too hot. Our forests stressed out too much during normal dry seasons. And then we put in these mechanisms where we did a better job with the mean annual cycle, but now we've made our porage too cold. We don't respond to drought. And so this is something that we're doing right now, is we're trying to work on mechanisms in the model, and it's more subtle now than it was, right? Previously we had this horrible mistake, right? We were way off what was observed. Now we're closer to observed, but it's harder to sort of get back to get to what's the actual signal that's coming out of those forests. But it is a place where we're working on the model, and we are showing improvements here. So in conclusion, we need these models for both evaluating our current system behavior and predicting the future of this sort of natural sink we have of half of the anthropogenic CO2 that we're burning each year. OCS and SIF are these novel, interesting things that allow us to observe nature in different ways. And by using them in our models, they give us new pathways to confront our models with data and evaluate how they're behaving. There is no silver bullet, right? When these new sort of features, 15 years ago it was carbon isotopes. People say, oh, it's going to allow us to figure the whole thing out. That just hasn't happened. But these things do give us new information, and they allow us to really make progress in our understanding. So really the value of models is in falsification. If I run a model and it matches an observation, whatever observation that is, if it matches it, I've learned nothing. Where I really get value from models is I run my model and it doesn't match what's observed. That now gives me the ability to create a hypothesis for why that's happening, to incorporate that hypothesis into my model and then attempt to falsify it. And this is the real place where models are valuable. And so things like carbonyl sulfide and fluorescence really allow us to do that. They give us new ways to falsify our models. And finally, frequently these results are unexpected. Like at Harvard Forest, it's indicating that our nocturnal stability calculation is erroneous. We didn't expect to find that. And so it's, sometimes it leads you off in interesting new directions. And with that, I conclude before I hit red with 30 seconds to spare. So, yeah, thank you.